Okay, so this is digestive enzymes in a little bit more detail. So first of all, looking at the digestion of carbohydrates and looking at the specific example of amylase. So amylase is a digestive enzyme that breaks down starch. The starch is a mixture of two polysaccharides. You've got amylose and amylopectin, each of which are made of long chains of alpha glucose molecules. So amylase works by catalysing the hydrolysis, the breakdown using water, the hydrolysis reaction that breaks glyosidic bonds in starch to produce maltose. So maltose is a disaccharide, which means it's got two glucose molecules, because di means two, a monosaccharide is things like glucose. Disaccharide means there's more than one, there's two. So amylase is produced in the salivary gland, which releases amylase into the mouth, and also by the pancreas, which releases amylase into the small intestine. There are also membrane-bound disaccharides, which are enzymes that are attached to the cell membrane of the epithelial cells in the lining of the ileum, which is the final part of the small intestine. And they help to break down the disaccharides, like maltose, into monosaccharides, like glucose. And again, this involves the hydrolysis of the glyosidic bond, so the splitting of that glyosidic bond using water. So here's the example of sucrose being split into alpha glucose and fructose. You'll have to excuse my poor drawing skills. So sucrose is a disaccharide, which will be broken down by sucrase, which will split this glycosidic bond using water to produce alpha glucose and fructose, which are both monosaccharides. I'll just say that again. Sucrase is a membrane, that should say bound disaccharide, and it catalyses the breakdown of sucrose, the disaccharide, into the monosaccharides, glucose and fructose. So the disaccharides maltose and lactose are broken down in a similar way to sucrose via hydrolysis of the glycosidic bond. So the disaccharide sucrose is broken down by sucrase into glucose plus fructose. Maltose, the disaccharide maltose, is broken down by maltase into the monosaccharides alpha glucose and alpha glucose. Lactose is broken down by the enzyme lactase into the monosaccharides glucose plus galactose, and it's all done by hydrolyzing the glycosidic bond. So the monosaccharides can then be transported across the epithelial cell membrane in the ileum via transporter proteins, which we'll look at in a different lesson. So now we'll look at the digestion of fats or lipids. So lipids are broken down by lipase enzymes, which catalyze the breakdown of lipids into monoglycerides and fatty acids. So this involves the hydrolysis of this time the ester bonds in lipids. So lipases are mainly made in the pancreas and then secreted into the small intestine where they actually act. So here's a triglyceride joined together by an ester bond, and then they are hydrolyzed, so two water molecules, into a monoglyceride, because you still have one of the glycerols left. One of the fatty acids left attached. So you get a monoglyceride and two fatty acids. So triglyceride has ester bonds. You use two water molecules which will hydrolyze two of those ester bonds to form a monoglyceride and two fatty acids. Just a little note about bile salts. Bile salts are produced in the liver and they do something called emulsification. They emulsify lipids. So they cause lipids to form small droplets. And bile salts aren't actually enzymes, but they are very important in lipid digestion. because if you take a big lipid globule and you emulsify it into smaller globules, by making the bits of fat smaller or the bits of lipid smaller, you increase the surface area, because as you know, the smaller the molecule, the larger the surface area to volume ratio. So if you increase the surface area, that means that there's more for the lipase enzyme to then act on. So it makes lipase's job a lot easier if the lipid globules are in smaller globules. So the formation of small droplets really does greatly increase the surface area of lipid that's available for lipases to work on. So you can see here that you've got a big lipid droplet, you add the bile salts and it emulsifies it into lots of little lipid droplets, which therefore increases the surface area. Once it's broken down into once the lipid has been broken down by lipase into monoglycerides and fatty acids, they actually all stick together with the bile salts to form a structure called a micelle. I think you might have heard of micellar water. So micelles help the products of lipid digestion be absorbed into the bloodstream. So micelles are literally little droplets made up of monoglycerides, fatty acids and bile, and they help with the absorption of the monoglycerides, fatty acids across the epithelial wall into the bloodstream. So now looking at the digestion of proteins. Proteins are broken down by different types of peptidases, so enzymes that catalyse the conversion of proteins into amino acids by hydrolyzing the peptide bonds between the amino acids. So you need to know about endopeptidases, exopeptidases and dipeptidases. So endopeptidases, the word endo means inside. That's why we have an endoskeleton, because we have a skeleton inside. So an endopeptidase actually cuts the polypeptide right in the middle. So it splits the peptide bond right in the middle of the molecule to form like kind of equally sized-ish molecules. So it splits the peptide bond right in the middle of the polypeptide. And some examples of endopeptidases are trypsin and chymotrypsin, which are both endopeptidases, and they're made in the pancreas and then secreted into the small intestine. Then you've got a pepsin, which is quite a well-known one, which is another endopeptidase, which is released into the stomach by the cells in the stomach lining. And pepsin's optimum conditions are acidic conditions, which is provided by the hydrochloric acid in the stomach. Then you've got exopeptidases. So as you know from the word exoskeleton, that means on the outside. So exopeptidases work on the outside of the polypeptide. They literally just chop off the very end amino acids. So exopeptidases work on the outside. They chop off the very ends of the polypeptide. They chop off little amino acids off the end. So single amino acids from the ends of the protein molecule. Last one you need to know about is dipeptidases, which are another type of exopeptidase that work specifically on dipeptides. They separate the two amino acids that make up the dipeptide by hydrolyzing the peptide bond between them. So dipeptidases are quite often found in the cell surface membrane of the epithelial cells in the small intestine. And obviously the products you get are two amino acids. And that is the end of digestive enzymes.